Assalamualaikum to my supervisor, Madam Farida Halim Muhammad Jafar, and to all my examiners, Dr. Ciputa and Dr. Asha. My name is Amira Kalma binti Shahrul Azri, and today I would like to present about my thesis, which is a comparative study of the essential oil components and antioxidant activity of Lavandula angustifolia with the commercial brands. First, introduction. Essential oils are the combination of saturated and unsaturated hydrocarbons or volatile compounds extracted from different parts of plants such as the seed, bud, flower, and the bark. There are several ways of extracting the essential oils from the plants such as cold press, hydrodistillation, and steam distillation, which are the most used method to extract. Some examples of essential oils that have therapeutic properties are lavender, which helps reduce anxiety and act as a stress reliever, lemon, which is for psychologically strengthening, and rosemary helps to enhance alertness and an energy booster. The problem statement. Naturally occurring antioxidants in the plants include flavonoids, tannins, phenols, and lignans are important for humans, which may help protect cells from damage that are caused by free radicals. Hence, this research on the essential oil focuses on its chemical components and antioxidant properties. The essential oil of Lavandula angustifolia extracted in the laboratory will be compared with those from only two commercial brands of Lavandula angustifolia oil, which are Young Living and Plant Therapy. Significance of study. The antioxidant activity of Lavandula angustifolia oil that will be extracted in the laboratory by hydrodistillation will be compared with the other two commercial plants of the oil. The activity of each oil will be determined by the chemical components in the oils. This study will give them the new knowledge on how important essential oils are as a natural antioxidants. The objectives of this study: first, extract the essential oil of Lavandula angustifolia by hydrodistillation. Compare the components of essential oils of Lavandula angustifolia extracted with those of the commercial brands. Compare the antioxidant activity of Lavandula angustifolia oil with those of the commercial brands. Literature review. First, description of Lavandula angustifolia plants. Lavandula angustifolia, commonly known as English lavender, belongs to the mint family of Lamiaceae and is known for its sweet floral scent. It is native to the Mediterranean and it is grown to be commercially produced as an essential oil that has cosmetic and medicinal uses. There are several species of lavender such as Lavandula angustifolia, Lavandula latifolia, and Lavandula intermedia. In this study, Lavandula angustifolia is chosen because it is readily available for purchase. The traditional use of Lavandula angustifolia, the Greek physician to the Roman army said that Lavandula angustifolia helped relieve indigestion, sore throats, headache, and also to clean wounds. Dried lavender tea can be made from its leaf, which has been told to help relieve insomnia. Essential oil components of Lavandula angustifolia. A study reported major components of lavender oil extracted by classical hydrodistillation, which are linalool, eucalyptol, linalool acetate, and borneol, whereas for microwave assist extraction, linalool, bifencyl acetate, linalool oxide, and lavandulol. Interestingly, the major components of the essential oil of Lavandula angustifolia may differ in some countries due to their environmental conditions, soil characteristics, harvest timing, and drying procedures. The composition of linalool differs in lavender oils of different origins. Antioxidants are components that can protect the cells against the free radicals that are produced in our body. A study from University of Life Science in Lublin reported that Lavandula angustifolia oil's antioxidant activity was observed due to the high concentration of linalool. Flower part of the lavender produce higher concentration of essential oil, mainly linalool, linalool acetate, phenolic, and anthocyanins, which contain greater antioxidant activity than the leaf part of the lavender. Now, methodology. First, preparation of the sample. 80 grams of dried lavender flowers were weighed and placed in the round water flask with 800 ml of distilled water as the solvent. And then the mixture was refluxed for 3 to 4 hours. The oil was collected after separating it from the aqueous layer. Anhydrous sodium sulfate was added to dry the oil, and the oil was stored in the vial and was kept in the fridge. Preparation of essential oils. 1.5 ml of each essential oils were diluted with 50% of ethanol. The mixture was diluted in 100 ml of volumetric flask, and 50% of ethanol was added up until the calibrated mark. To determine the total phenolic content, first 1.5 ml of diluted essential oil was diluted with 3 ml of distilled water in the test tube. 1.5 ml of diluted essential oil was diluted with 3 ml of distilled water in the test tube. Then 0.2 ml of Folin 3 agent was then added and mixed together. 1 ml of sodium carbonate and 3 ml of distilled water were 1 ml of 20% sodium carbonate and 3 ml of distilled water were then added, and the mixture was allowed to keep at room temperature for 2 hours. The absorbance was then measured at 760 nanometer. PPH assay. The PPH reagent was dissolved in ethanol. 1.5 ml of diluted essential oil was added into 1 ml of the PPH solution, and then the samples were incubated in the dark for 10 minutes, and the absorbance was measured at 517 nanometer. Next, result and discussion. The chemical composition of the essential oil was determined by GCMS, and a total of 12 compounds were identified. There are 5 major compounds in the extracted oil, which are linalool, alpha-terpineol, terpinfool, lavandulic acetate, and guanylyl acetate. A study by Danila et al. 2018, they reported linalool as the major compound in Lavandula angustifolia. Therefore, it can be suggested that the antioxidant effect of Lavandula angustifolia extracted oil in this study is due to its major compound, linalool. For young living, a total of 8 compounds were identified, and the major components are linaloic acetate, linalool, caryophyllene, cis-B-farnesene, and lavandulic acetate. The brand Plant Therapy Essential Oil reported 13 chemical compounds, and the major components of its oil are linalool, linalyl acetate, terpinfool, trans-B-ocimene, and lavandulic acetate. The data showed both Plant Therapy oil and Lavandula angustifolia extracted oil contain linalool as the major compound in their oil composition, whereas Young Living has linalyl acetate 37.21% as its major compound. According to Kitam et al. 2016, linalool has been shown to have antibacterial, anti-inflammatory, anti-cancer, and antioxidant effects, and the high linalool composition was believed to contribute to the oil's antioxidant properties. Higher composition of linalool in Young Living oil means higher quality of the essential oils, which may suggest it's more expensive compared to Plant Therapy oil. Lavandula angustifolia extracted oil was observed to have the highest percentage of linalool, 38.5%, thus suggesting that it has better antioxidant activity compared to the 2 commercial brands oil. In addition, we also observed that all 3 essential oils have different major compounds with different composition detects. This may be due to different methods of extraction used, which were hydrodistillation, steam distillation, and cold press method. Determination of total phenolic content. Shown in the table, Lavandula angustifolia has the highest phenolic content, which is 156.85 microgram gallic acid equivalent per ml among the 3 oils. Compared to a study by Dudal et al., polyphenolic compound in samples from 3 years old Lavandula angustifolia plants range from 12.44 to 18.16 mg TAE, which was the lowest compared to the other Lamiaceae family. Total phenolic content of Young Living essential oil is 13.55 microgram gallic acid equivalent per ml, and the lowest TPC was found in Plant Therapy essential oil, which is 12.53 microgram gallic acid equivalent per ml. Lavandula angustifolia extracted oil and Young Living essential oil are expected to perform well in antioxidant and antibacterial activities due to the higher phenolic content in their oils. TPPH radical scavenging activity. The half inhibition concentration of Lavandula angustifolia extracted oil and ascorbic acid are 464.63 microgram per ml and 822 and 57 microgram per ml respectively. Young Living essential oil has higher radical scavenging property compared to Plant Therapy essential oil. This shows that Young Living has stronger antioxidant activity with 942.99 microgram per ml than Plant Therapy essential oil, which has weaker antioxidant activity with the IC50 of 975.67 microgram per ml. Overall, Lavandula angustifolia extracted oil has the strongest antioxidant activity compared to the other 2 of commercial brand essential oils. In conclusion, from those analyses, the common compound from all 3 essential oils is known as linalool, which may contribute to the antioxidant activity of the oils. The Lavandula angustifolia extracted oil showed the highest TPC, which is 156.85 microgram GAE per ml, and Young Living has higher TPC, which is 13.55 microgram GAE per ml compared to Plant Therapy oil with a 12.53 microgram GAE per ml. From the PPH assay, Lavandula angustifolia extracted oil showed the strongest antioxidant activity with the IC50 of 464.63 microgram per mil, whereas plant therapy has the weakest antioxidant activity with IC50 of 975.6 microgram per mil. IC50 of young living is 942.99 microgram per mil, which showed the higher antioxidant activity than the plant therapy oil. Lavandula angustifolia extracted oil can be concluded to have good antioxidant property, which can be used to protect our cells from the damage caused by free radicals. Other than that, higher composition of linalool in young living oil means higher quality of the essential oil, which may suggest it's more expensive compared to plant therapy oil. To get a better result, we should have conducted the experiment carefully by following each steps properly without any mistakes. Thank you for watching and thank you for listening.